Okay, welcome to Chapter 4. This week we're going to be talking about the cell. Well, we won't just be talking about human cells, we'll also be talking about what bacteria, archaea, fungi, protists, and plant cells look like. So let's start off as an introduction to cells. So in our first week we kind of talked about the different types of general cells, the prokaryotes and the eukaryotes. Cells are the smallest units of living things. They make up our tissues, which make up our organs, which make up our organ systems, which make up us. Sometimes they make up an entire organism by themselves, as some organisms are unicellular, and that is the only thing that runs them, like bacteria. Some organisms are multicellular, like humans and plants and fungi and a lot of other organisms. Humans have several trillion cells. So going forward, we'll talk a little bit about the differences between prokaryotic cells and eukaryotic cells, and the organelles that will be making up these cells, which will impact their function. So first, how do we see these cells? Most cannot be seen with the naked eye. There are a couple of exceptions. The ovum that females expel when they ovulate, that can be seen with the naked eye. It's one of the largest cells in the human body. But most cannot be seen with the naked eye. So in order to be able to see them, we're going to need to be using microscopes, microscopy. They magnify and invert images as shown here below with that E. It's magnified and inverted. The two main types of microscopes are light microscopes and electron microscopes. Light microscopes are good for viewing living organisms. These are typically the student type of microscope you'll see in a laboratory setting. Usually the slides that you'll be viewing will be stained, so the organisms are no longer living, but that staining is needed to be able to discern between the different structures within the cell. The total magnification of a light microscope is 400x. That's not going to be the same as the objective lenses that you'll be looking through. On the other hand, electron microscopes will use a beam of electrons rather than a beam of light to magnify an image. It's not beneficial for viewing living things. You will not be able to use one of these in a typical student laboratory because they are so expensive and also so giant. But they do provide a much greater magnification and resolution for looking at images. So we can see some examples here. We're looking at red blood cells in blood vessel walls. So you can see that as we go up in magnification from 250x to 10,000x to this scanning electron micrograph, you can see these red blood cells in much greater detail than with your typical light microscope. So how did we even get to this place where we were able to start looking at cells through microscopes? Well, the first scientist who looked through a lens to see single-celled organisms was Antony van Leeuwenhoek in the 1600s. And once he saw these single-celled organisms, he called them animalcules, which is a pretty cute name, and I think we should stick with it. Next is Robert Hooke in 1665. He coined the term cell based on the appearance of cork. It kind of looked like a cell and like a prison cell. And then Schleiden and Schwann in the 1830s developed a unified cell theory, which was contributed to a lot by other scientists throughout the years as well. But the unified cell theory states that one or more cells comprise living things. The cell is the basic unit of life, and new cells arise from pre-existing cells. So the main distinction between prokaryotes and eukaryotes. Prokaryotes are found within bacteria and archaea. Eukaryotes are found within plants, animals, fungi, and protists. For the word prokaryotes, pro means before and kary means nucleus. So bacteria and archaea do not have an actual nucleus. There's no membrane-bound place for the genetic code to sit. In eukaryotes, U means true, so they have a true nucleus, and they have a membrane-bound organelle that holds all the genetic information. The components of the prokaryotic cell include the plasma membrane, a cytoplasm, DNA, which is found within a nucleoid, which is similar to a nucleus, but it's not membrane-bound, and ribosomes. So they lack a nucleus and membrane-bound organelles. And what's important about membrane-bound organelles is that they have a distinction between themselves and the cytosol. So whatever within the cytosol or the cytoplasm free-floating around, it can come in contact with any of the organelles and disrupt their processes. Sometimes prokaryotes contain a cell wall and a polysaccharide capsule. So with your discussion for this week, it's a really cool discussion. You'll be looking at bacteria that have these polysaccharide capsules. Capsules increase the virulence of a bacterial strain, meaning that it's basically a harder bacteria to get rid of from a host. And that's because these capsules make them a lot more difficult to fight off. Some also contain a slime wall or a slime mold, and those are also very cool to look at. Some prokaryotes also have fimbriae. Fimbriae are these little structures that allow the bacterium or other prokaryotic structure to be able to attach to host cells, and it's kind of like Velcro. Flagella are these long whip-like extensions from the cell. It's flagellum if it's singular, flagella if it's plural, and it's used for locomotion. And then they also have pili or pili that extend. They're kind of like hair-like extensions that come off of the cell. Those are used for the exchange of genetic information during conjugation, which is bacteria's form of reproduction. Prokaryotes are smaller than eukaryotic cells, which allows for this quick diffusion of ions and organic molecules. Waste can also quickly diffuse across the membrane. The smaller the cell is, the easier for the plasma membrane to the more efficient the flow of ions and nutrients and waste across the plasma membrane. So a smaller cell size increases the efficiency, there's more surface area, and the spherical shape is really beneficial as well. So the eukaryotic cell has a membrane-bound nucleus, which has several chromosomes within it, membrane-bound organelles, plasma membrane, and a cytoplasm. So it's pretty similar, except it has these membrane-bound organelles. It also has non-membrane-bound organelles, and the nucleus is the main difference. So the suffix L means little, so organelles are little organs within the cell that makes it run. Kind of think of it like you're the cell, and you have organs, and your organs are helping you run. Organelles are helping the cell to run. Many get this confused with something that's larger than a cell, so just keep that in mind. So the first component of the cell that we're going to talk about is the plasma membrane. So the plasma membrane is a phospholipid bilayer. Bilayer meaning that there's two layers. There's a hydrophilic component and a hydrophobic component. Hydrophobic meaning water-fearing, hydrophilic meaning water-loving. So the outside is going to be the water-loving component, inside is going to be hydrophobic, and then it'll be hydrophilic on the inside of the cell. So this will help to control all of the passage of organic molecules, ions, water, oxygen, and also excreting waste from the inside of the cell. And the plasma membrane is so complex that we're going to spend an entire chapter talking about it next week. Some plasma membranes contain microvilli, which are these finger-like projections that come off of the surface of the cell. They kind of have these invaginations that go up and down. It helps to increase the surface area, and these are generally found in cells that are specialized for absorption of nutrients. So you'll find a lot of these cells in the in the linings of your intestines and your stomach lining to help absorb these nutrients as they come in from your food. There is some implications with this with celiac disease. So with celiac disease, the immune response from that disease is going to damage these microvilli and make the body unable to absorb nutrients. So it's not even just when people have celiac disease that they are in pain and they have these digestive symptoms. It's that they can't absorb the nutrients that they're taking in, any of the nutrients, not just the gluten, everything. So it's a broader issue than just I can't eat gluten because, I have symptoms related to it. It's if they eat gluten, they won't be able to absorb anything they digest. And that can lead to a lot of issues. And over time, this can even lead to cancerous changes in the tissue if they continue to take in gluten with this disease and with the immune response they get from it. So once within the cell, once we pass this plasma membrane, we come in contact with the cytoplasm or the cytosol. They're pretty much continuous with each other. The cytoplasm is more of a jelly-like material. The cytosol is more of a fluid. It's really just a difference in consistency. The cytoplasm itself consists of about 70 to 80% water. It's the site of many of the metabolic reactions that are going on in your bodies, or in plants, or bacteria. And that semi-solid, jelly-like consistency comes from a lot of suspended proteins. The nucleus is the largest organelle in the animal cell. It's going to house all of the DNA, and it directs the synthesis of ribosomes, and this sign means therefore, and therefore proteins. So within the nucleus is this nucleolus. I know they look very similar, because especially when they're in the plural form, because it's nuclei, nucleoli. But the nucleolus is responsible for making up the ribosomal subunits. And ribosomes make proteins. So the nucleus directs the synthesis of proteins. It also has all the genetic information to tell RNA how to make proteins. Basically, it has the instruction manual. The nucleus is surrounded by this nuclear envelope, which has a lot of nuclear pores. As you can see, these little holes in between it. DNA is too large to be able to leave through those nuclear pores, but RNA is just small enough that it can enter to be able to get that instruction manual to start the process of making proteins. Here on the outside, what you see is the endoplasmic reticulum, which we'll talk about in a few minutes, but it is directly continuous with this nuclear envelope. So the nuclear envelope is also a phospholipid bilayer. Like I said, it contains these pores to help control the passage of ions, molecules, and RNA. And it also has its kind of own cytoplasm within the nucleus. It's called nucleoplasm. And it contains the chromatin and the nucleolus. The chromatin is just going to be your genetic information, all wrapped together with some proteins. It's loosely bound, and right before your cells start to divide, they become tightly bound together to form chromosomes. So chromosomes are made up of DNA. In prokaryotes, DNA is circular, but it's linear in eukaryotes. The number of chromosomes will vary per species. For example, fruit flies only have a couple of chromosomes. We have 46, or 23 pairs, one from each pair from each parent. The chromosomes are only visible during cell division. Any other time, they are visible as chromatin. So like we said, the nucleolus is within the nucleus. It's going to make rRNA or ribosomal RNA and assemble the ribosomal subunits. Those are then transported from the nuclear pores to the cytoplasm, where the ribosomes can assemble and become free-floating or fixed. and when they are fixed to the rough endoplasmic reticulum. So ribosomes, like I said, are responsible for protein synthesis. The first step of protein synthesis is transcription, so it's going to facilitate that transcription of DNA to mRNA. And that's because the DNA cannot leave the nucleus. We're going to need to make it into a smaller form. So it's kind of like the RNA goes into the nucleus and sees the instruction manual, jots down a couple of notes, and leaves with that because it can't leave with the giant instruction manual. And then the mRNA travels to the ribosomes where translation will occur, and the translation will take that instruction manual and transform it into amino acids, which will come together to form the primary structure of proteins. Ribosomes can be in clusters, and usually when they're in clusters, they are attached to the rough endoplasmic reticulum, or they can be free-floating in the cytoplasm. They will have large and small subunits, and they will be found in practically any cell, but mostly in those that are going to synthesize more protein. So you can imagine that the tissues that are making a lot of hormones, they're going to have a lot of ribosomes. Those that are making a lot of enzymes are going to have a lot of ribosomes. Skeletal muscle will have a lot of ribosomes, and so on. Mitochondria, I'm sure you've heard, is the powerhouse of the cell. It forms ATP, which is the form of energy that we use in order to do any of our day-to-day processes. ATP stands for adenosine triphosphate. Any of the organs or tissues that require more energy will have more mitochondria in their cells. So, like we said in the beginning, structure impacts function. So the more mitochondria, the better that they can function in creating more energy. Mitochondria contain their own ribosomes and also their own DNA. That's pretty crazy. So there's actually entire diseases that are based upon mutations in mitochondrial DNA. And mitochondrial DNA is passed on strictly maternally. So any of your mitochondrial DNA you got from your mother. The inner folds of the mitochondria are known as cristae, and they're found within the inner mitochondrial membrane because the mitochondria is a double membrane. And between the membranes is the mitochondrial matrix. Both of those places are sites for this creation of energy through glycolysis, through the Krebs cycle, and through the electron transport chain. Peroxisomes are going to carry out oxidation reactions. So peroxisomes are extremely important because you have these free radicals in your body that are formed through the breakdown of these fatty acids and amino acids from the things that you eat. So free radicals, if they're left to do what they do best, they will damage tissues, and they can even cause mutations, and they contribute a lot to aging. So peroxisomes are really important for detoxifying both free radicals and poisons in the body. And they're called that because one of the main free radicals that is formed from the breakdown of fatty acids and amino acids is hydrogen peroxide. So peroxisomes, like peroxide. So the liver has a lot of peroxisomes because it does a lot of the detox in your body. Plants have a similar structure, but they're called glyoxysomes, and they convert fats to sugars. They have a completely different function, but they are similar in structure. Vesicles and vacuoles, very similar, but they have different functions. So they're both membrane-bound sacs for storage and transport. Vesicles are much, much smaller. They tend to fuse with the plasma membrane and transport materials to different organelles. They can transport materials to the plasma membrane to be excreted by the cell, or they can even bring in materials from the extracellular environment to an organelle for processing. Vacuoles are much larger, and they're mostly used for storage. So the best example is the central vacuole in plants that is used to store fluid and it kind of keeps the cell, helps to maintain the structure of the cell wall. Without this central vacuole being filled with fluid, the plant will wilt. The endoplasmic reticulum is a series of interconnected tubules and sacs. It's directly connected to that nuclear envelope. It modifies proteins, synthesizes lipids. It also has some detoxification functions as well. The hollow inner portion of the endoplasmic reticulum is known as the lumen, or the cisternal space. And it can be broken up into the rough endoplasmic reticulum, or the rough ER, or the RER, and the smooth ER, or the SER. The rough endoplasmic reticulum is called such because it is studded with ribosomes. So those fixed ribosomes stud the surface. It's also responsible for modifying proteins that are transported by the ribosomes, makes phospholipids, and then any products that the rough ER makes are transported to their destinations by those vesicles that we just talked about. The smooth ER is continuous with the rough ER and does not contain ribosomes, hence the name smooth. It synthesizes carbohydrates, lipids, steroid hormones. It's more of the detoxifier of the two, detoxifies medications and poisons, and it stores or sequesters calcium ions, so you might find a lot more smooth ER in muscle cells, because muscle cells use calcium to be able to contract. The Golgi apparatus is known as the post office of the cell because it sorts, packages, and tags products from other organelles to be sent elsewhere in the cell or out of the cell. There's two different phases of the Golgi apparatus, a cis phase and a trans phase. So, products that are entering the Golgi apparatus to be sorted, packaged, and tagged will fuse with the cis phase via a vesicle and empty its contents. The Golgi apparatus will do what it does best, and then those products will be excreted via the trans phase to either be excreted by the cell or to travel somewhere else within the cell. So an example of something, of a structure that would have a lot of Golgi apparatus would be the salivary glands, as they make a lot of products to be excreted in the form of saliva. Okay, so now we're going to go into more of what is within an animal cell and what's within a plant cell. Everything we've talked about so far is within both. So, centrosomes are only in animal cells, lysosomes are only in animal cells, cell walls are only in plants, as they are chloroplasts and central vacuoles. So, centrosomes, only in animals. It's known as a microtubule organizing center, or an MTOC. It contains a pair of perpendicular centrioles, and it replicates itself right before cell division. So it helps with some of the structural framework of the cell, and it also plays a role in cell division, though scientists are still trying to kind of figure out what exactly its role is. Lysosomes are known as the garbage disposal of the cells. These are not found in plants. In plants, vacuoles take on this responsibility, but lysosomes have these acidic enzymes to help destroy pathogens. So you might find a lot of these in white blood cells that help to destroy some of the foreign invaders that are in our bodies in the form of bacteria and viruses. Cell walls, only in plants, help to protect the cell. It helps to provide structural support. It gives shape to the cell. Some fungi and protists also have cell walls, and they're typically composed of peptidoglycans and cellulose. So that's why when you bite into a vegetable that's fresh, generally you'll hear a loud crunch, and that's because of it being so high in cellulose and peptidoglycans. You're breaking down the cell walls of these plant cells. Chloroplasts are going to be the structures within plants that help to carry out photosynthesis. So the use of carbon dioxide, water, and light to make glucose and oxygen. Chloroplasts also contain their own DNA, kind of like mitochondria, and ribosomes. Plants are known as autotrophs because they make their own food. Animals are heterotrophs because they have to take in other organisms in order to make energy. The central vacuole in plants occupies most of the cell's area. It regulates the cell's concentration of water due to the environment, so there might be some specializations of this vacuole in plants that are in more dry environments or cold environments. When plants wilt, it's because this vacuole is not completely filled with liquid, which reduces the support of the cell wall. The cytoskeleton provides a framework. This is within plant and animal cells. The cytoskeleton is made up of microfilaments, intermediate filaments, and microtubules. Microfilaments, as the name suggests, are the smallest of the cytoskeleton components. They're also known as actin filaments. They're composed of two globular proteins that are intertwined, which are actin, and they serve as a track for movement of myosin, which together form muscle contractions, and they also help to provide rigidity and shape to cells. Intermediate filaments, such as keratin, provide more of a structural framework rather than playing a role in cell movement. Intermediate filaments help to withstand tension, help to maintain the shape of the cell, and anchor the nucleus and organelles in place. Microtubules are the largest of the cytoskeleton components. They help to resist compression. They provide a track for vesicles to move. They pull replicated chromosomes to their poles during cell division. And they are the structural elements of cilia, flagella, and centrioles. Flagella, like we've already talked about, are these whip-like extensions off of the cell. They extend from the cytoplasm. They're made up of a 9+2 microtubule array. It's just the way that the microtubules are arranged to form this flagellum. Cilia also have this 9+2 microtubule array. In humans, there's only one type of cell that has a flagellum, and that is the sperm. Cilia extend from the entire plasma membrane surface. They're tiny hairs. They're found on a lot of the tissues in our body, specifically that of the fallopian tubes to help bring in the egg and transport it to the uterus to be implanted. It's also found in the respiratory tract to help to bring up dirt and debris that you might have breathed in. If you are a smoker, you might have paralyzed or completely damaged the cilia in your respiratory tract and made it more difficult to be able to cough up that debris. So these are some pretty good tables to look at when you're studying these organelles. The extracellular matrix is composed of proteins and proteoglycans. Most abundantly is collagen. They also contain protein receptors. Different chemical signals can turn genes on and off. One really good example of that is blood clotting. So when the blood vessel wall is damaged, it shows a protein receptor which stimulates platelets to aggregate and to help form a clot. Lastly, how do cells communicate with each other? In plants, they communicate via Plasmodesmata. So neighboring plant cells do not typically touch because of these bulky cell walls. So the plasma membranes of neighboring plant cells contain these channels called Plasmodesmata to help connect the cytoplasm and communicate. Water and soil is transferred through roots via the vascular tissue xylem and phloem. In animal cells, we have tight junctions, which are watertight seals between animal cells. They're held together by the proteins claudins and occludins. They're found in epithelial tissues of the organs and skin, which helps to maintain that waterproof barrier of our skin. If our skin wasn't waterproof, then we'd be flooded with water and our cells would burst and we would burst. It helps so that urine does not leak out of your bladder through the cell wall or through the gaps in cells. Desmosomes are like spot welds between animal cells. The proteins that they're made up of are called cadherins. They're also found in organs and tissues that stretch, such as the skin, heart, and muscles. Gap junctions are similar to Plasmodesmata, but in animal cells, so they're channels between animal cells connecting their cytoplasms. They help to transport ions and nutrients. And they have a lot of importance in cardiac and muscle cells to allow for electrical impulses to travel. The proteins that they're made up of are called connexins. So think of all of these as like the way that the bricks are laid together. These different cells are connected to each other by one of these different cellular connections, and it will vary depending upon their function. So if you have any questions, please let me know, and good luck on your test next week.